Good afternoon, my name is Dominic Toledo, the boy on the corner of the image. I am a member from the Team 5 from class 205, and today I will talk about the field of study of biology and its branch. First of all, fields. Biology is a science that studies life from different standpoints, from the structure of atoms and molecules to the biology of evolutionary development of population. Now, the branches. Biology has a lot of branches, but one of the most important ones are anatomy, anthropology, biomedicine and biochemistry, and botany. We also have embryology, cytology, genetics, histology, immunology, virology, and zoology. Now, let's talk about the second topic I will explain you today. How life started in the planet. Even today, humanity don't have an accurate answer to that question, but this is the most close answer to it. First, we have to ask us this question. What factors favored the creation, assembly, and replication of the first molecules? During the whole life of being a civilization, we have had different beliefs about this. First, we talk about that we were created by a god, also a expected opinion, or even that we were created just spontaneously. With the time, we try to get a new answer to that question. We try to make a new explanation to it. Some consider that the electrical activity in the atmosphere originated life. Other people suggest that the creation of life occurred in the volcanoes. Some more say that the origin of life was on the bottom of the oceans. And some others think that the living beings arrived transported by meteorites or comets. The only thing that we actually know is that everything that has come before us gave rise to a system. Which system? The DNA and the RNA. Knowing all this, even knowing all of the DNA and being able to search on it, we haven't been able to know what is the origin from life. The importance of this question is not its answer. The importance of it is that we haven't seen the end of this story yet. We haven't seen the... So, what we have to do now is to evolve to be able to answer the question. And that question is the topic that I had explained. How did life originate on the planet? With that said, I'll leave you with my partner.
History of the microscope, the cell, and the cell theory. History of the microscope. The observation of the world of tiny things began with the investigation of the magnifying lenses, which are a very rudimentary version of the microscope, now known as the composite microscope, because it combines more than one magnifying glass to see objects and shows them in increased sizes. The invention of the oldest magnifying lens took place in the Syrian Empire in 750 before Christ. Also, the Greek, Egyptian, Roman, and Babylonian civilizations manufactured and used magnifying lenses. In Italy, artisans began to carve magnifying glasses that were used as spectacles. Gradually, this use of lead manufacturers to improve and refine their carving techniques, which culminated in the invention of transcendental instruments such as the telescope and the microscope. Who invented the compound microscope? Perhaps Zacharias Janssen, Galileo Galilei, or Cornelius Drebbel were the inventors. What we can say is that the microscope was invented at the end of the 16th century and the beginning of the 17th. The first person who names this invention as the microscope was Giovanni Faber in 1625. According to the records of the time, the magnification range achieved by the microscope was between 3 to 9 times increased the size. This type of microscope had the basics, one lens that worked as an objective and the other as an eyepiece. Who was able to glimpse its importance and use it for scientific purposes was Robert Hooke in 1665. In modern microscopes, illumination is performed by a light bulb and a condenser. Anton van Leeuwenhoek developed a new technique to manufacture lenses that allowed him to observe samples with magnifications up to 200 times. He published his results in 1684. It consisted of a single lens, but its main manufacturing technique was the secret to achieving magnification ranges never reached before. In the 19th century, it was possible to perfect the microscope, and it became a handy tool, especially for the medical field. The history of the cell. The official story begins with Robert Hooke, who in 1665 published the observations he made with the microscope he created. Hooke observed the bark of the cork oak and noticed that the tissue was made up of small spaces which repeated themselves like the cells of a honeycomb. He gave to these spaces the name of cells, from the Latin cellulae, that is, little chambers. What Hooke observed were dead vegetable cells, so the description of the cell content had to wait some years until Anton van Leeuwenhoek, with the lenses that he crafted, observed and described in more detail cells such as spermatozoa and protozoa, and even some bacteria. Although the research continued for many years, it was not until 1830 that Theodor Swan and Matthias Schleiden proposed what later became the first postulate of the cell theory. The 19th century was abundant in cellular discoveries. Robert Brown described the nucleus. Johannes Evangelista Purkinje introduced the terms protoplasm. That is, he was the first to describe the cellular cytoplasm. Rudolf A. von Kolliker identified the mitochondria, and Rudolf Virchow observed that the cells come from pre-existing cells. The work of Louis Pasteur on the metabolism of yeast and asepsis cleared the way to the progress of research. August Weismann discovered that both current and ancestral cells are similar in structure and composition. In 1981, Lynn Margulies proposed the endosymbiotic theory, which explains with solid arguments the origin of eukaryotic cells. Cell theory. Many researchers participated indirectly to generate the postulates of the cell theory. However, 3 of them are recognized as authors: Schwann, Schleiden, and Virchow. The postulates of the cellular theory affirm that the cell is the morphological and structural unit of all living beings. In other words, all living beings are made up of cells and the products these cells generate. Omni cellula ex cellula, which means every cell comes with a pre-existing one. This postulate debunks the theory of spontaneous generation. The cell is the physiological unit of life. All vital functions take place within the cells, and only one is enough to generate life. The cell contains the heredity information for the control of its development, growth, reproduction, and to transmit that information to the next generation.
I'm going to talk first about the cellular transport. The cellular transport occurs on the cell membrane when the cell receives nutrients and hormones from outside the cell. It could be from active way or passive way. Active is when the cell spends energy, and passive way is when the cell doesn't spend energy. Then we could see that this has a simple definition, is when the components pass through a filter by itself and rearrange into the cell. Then the facilitated diffusion is when this depends on some proteins. Then we can talk about the endocytosis. Is a incorporation of material into the cell. The exocytosis is the secretion of the material produced of cells. Then we are going to pass from to the other subject, the levels of organization. In the levels of organization, we could see that the first level is the atom and the last is the biome. We could see that these scales in atom, molecule, cellular organelles, cell, tissue, organ system, organism, population, community, ecosystem, and it ends in the biome.
Homeostasis. Claude Bernard observed the stability of physiological parameters such as body temperature, heart rate, and blood pressure, and in 1865 he called it homeostasis. Each cell will take care of its self-regulation, that is, maintaining its homeostasis. The term homeostasis comes from the union of two Greek words, which in Spanish are translated as homeos and stasis. Homeos means equal or similar, and stasis means state or stability. This preservation of internal conditions is achieved thanks to metabolism. An example is the regulation of body temperature. Also, an excellent example of negative feedback is the maintenance of homeostasis of the blood glucose concentration. The insulin stimulates the entry of glucose into other cells and its preferential use, and also the glycogenesis in hepatocytes and muscle cells. When there is not enough glucose in the blood, the alpha cells of the islets of Langerhans respond by releasing glucagon. Glucagon stimulates glycogenosis, and when glucose levels are regulated, alpha cells stop releasing glucagon.